So what we see here are two kidney epithelial cells, and this cell up at the top is going to stay in interphase. It's not going to be dividing, but this cell down here at the bottom is getting ready to undergo mitosis. And so we're going to go through this a couple of times, but I want you to pay attention to what's happening here in the nucleus. Again, we're seeing that the histone proteins that are complexed with the DNA in chromatin are labeled with fluorescent orange, and the microtubules are labeled with fluorescent green. And so you're going to see, as this cell enters mitosis, the highly compacted form of DNA starting to form, so those chromosomes that are highly compacted becoming visible. And you're also going to see that this cellular microtubule network gets massively reorganized by these centrosomes that are present right here at the nuclear envelope. So let's watch this a couple times, and I want you to pay attention to what happens with the DNA, because we're going to see something very interesting. Okay, so let's watch that again. I'm going to pause it at key points here. So we've seen the chromosomes condense here. You've seen the microtubules get reorganized. And what we're seeing at this stage is that the microtubules are starting to form their attachments to the kinetochores and starting to tug on these chromosomes. And that's why they're wiggling and wiggling and wiggling. And you can see pretty early on that the chromosomes get connected up correctly, so they're being tugged from opposite directions. But look at this guy down here. You see what's happening with this DNA molecule? It's not connected up correctly. It's being pulled only from this centrosome, but not from this centrosome. And so the cell actually has to wait for that DNA molecule to get connected up correctly before it proceeds to anaphase and telophase and the rest of cell division. So we'll watch it one more time. So tugging, 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 microtubules connecting up to kinetochores, and motor proteins pulling, pulling, pulling. And everybody's getting connected up and pulled to the midline of the cell equator, but the cell doesn't divide yet because that chromosome was not connected. As soon as it gets connected up and tugged to the middle, the cell gets the signal that equal tension is present, all chromosomes are correctly connected, and the cell proceeds to anaphase.